Hi, my name is Nadezhda, and as I said, I'm going to talk about the novo full-length transcriptome for Lepeophtheirus salmonis. A good transcriptome is an important tool to study which cell processes occur at a specific time in an organism. And one can find different tools for create a transcriptome, like for example use genome sequencing data to predict transcript sequences, use second-generation sequencing data producing short reads, and here is a short overview of the principle. We start with mRNA molecule that is reverse transcribed into cDNA, randomly fragmented into many molecules, amplified, sequenced, and assembled into transcripts. In addition, the third-generation sequencing has opened the possibility for long-read single mRNA sequencing. The currently available transcriptome for salmon lice are generally based on first two methods, but disadvantages with those strategies are genosiform identification difficulties and possible errors with short reads assemblies like repetitive sequences and gaps in sequencing coverage. What about the third-generation sequencing? I'm going to talk specifically about the PacBio sequencing that can sequence the full-length mRNA molecule, avoiding short reads assembly problems. Another important advantage of this method is possibility for identification of different splice variants. And here's a short overview of the method. We start again with an RNA molecule, which is reverse transcribed into cDNA. It's prepared as a multi-bell library. Primers and DNA polymerase binds to the sequences. Molecule are further circularized and sequenced in several times. What's the main difference between second and third generation sequencing? Both methods starts with reverse transcription of mRNA molecule to cDNA, but in PacBio, the molecules are not further fragmented and are sequenced in full length without amplification, what can result in relatively higher error rate and lower throughput compared to Illumina. So, to avoid these disadvantages, we have used both methods on the same samples for provide a hybrid assembly. And here you can see an schematic illustration of alignment of PacBio and Illumina results, where we can find an error in PacBio transcript that was corrected by short reads from Illumina. And in addition, we can find a gap in Illumina results, what could be a problem in assembly without the long full-length PacBio transcript. The background of our project is a study by Sigmund Ramberg et al., where they generated a de novo transcriptome for Atlantic salmon. And to cover differences in gene expression during life stages, we are going to generate two transcripts, one for adult female parasite and second one for its progenies in the copepodid stage. And you can see an illustration of gene differences during life stages in LS NAV 1.3 gene, previously presented by Sine and Julie from my group. Using this assembly method, it becomes possible to avoid a short reads assembly problems and identify novel full-length splice variants, even they are many kilobase long or show a low expression, and create a high-quality transcriptome, a valuable resource for further salmon lice research. Thank you for watching.